Okay, hello. So we're going to finish up the rest of our guided notes. Okay, starting here at the genetic code. This is just under the exons and the introns part. Okay. So remember, proteins are made by joining amino acids. This sentence right here, you're going to see all the time right here. This is a super important sentence. Okay? So the amino acid sequence influences the shape of the protein, therefore influencing the shape of, the function of the protein. Okay? Remember, shape is all that matters in the body. It's what makes things happen. So, okay, we know what a codon is. It's 3 consecutive nitrogenous bases. And each codon is going to correspond to 1 amino acid. Okay. Okay. Absolutely nothing that you need to memorize here, but there are 64 different codons. Okay. AUG is the start. You probably know that by now because you've repeated it so many times. And these are all the stop codons. Also, don't need to memorize it. You'll always have the chart. This circle chart right here, same information, but just organized a different way. It's going to be bigger in a second where we can actually look at it. Okay. This sentence right here, super important. Okay. The genetic code, aka DNA, is universal for all organisms, meaning every organism that exists has DNA, and it's the same kind of DNA, meaning it's made up of A, C, G, and T, or U, and when we get into RNA. Every organism uses these 4 letters, and somehow we have the great divide we see between us and bacteria. Even though we use the same 4 letters, they're in way different sequences, and yeah, so we have different organisms, but they all have the same DNA. We'll get more into that a little bit later when we talk about evolution. But like it says right here, this is part of the evidence that says that all organisms have a common ancestor down here, right? And you guys have seen Tree of Life before. It looks something like this, blah, blah, blah. But the point is that we all start at one place. There's one common ancestor. And we can point to the fact that we all have A, C, G, and T for that. Okay, so here's the circle chart. You read it the same way. In here, this inner circle, the big ones, that's going to be your first letter in your codon. The next one's going to be your second letter, and this one's going to be the third letter. So if you wanted to look up GUA, it'd be GUA. So that would be valine. All right, so this is just asking you to translate these codons. You can do that by yourself. I'm just going to flip through those really quickly. All right, translation. So we know is the process of decoding the mRNA into the amino acids and thus the polypeptide. Again, here's the same sentence again. The sequence of nucleotide bases in the mRNA gives the order of amino acids, which shows how the polypeptide is going to turn out. Okay, so on to translation, the first step. The ribosome is going to attach to the mRNA at the start codon. All right. Got it. Then the tRNAs start coming in. Remember, they have an anticodon at the bottom, and the mRNA has a codon. All right. It's important that they're unpaired bases because that's the only reason why they connect. Again, because of shape, because of polarity, because of hydrogen bonds. If the tRNA had at the bottom it had, I don't know why this is tickling me so much, but if it had AUG and it was paired up, so it had UAC, it wouldn't want to do anything. It would never come and connect to the mRNA. So that's the reason they're unpaired. Okay, so this is just a picture of transcription and translation, the whole thing in an example. Cool, you guys already know how to do this for sure. All right, so going on to the next part of translation, this is where we talk about the tRNAs actually coming in. The anticodon attaches to the codon temporarily via a hydrogen bond. Remember that's the bond between the bases. It is a weak bond. Okay, and we have an amino acid at the top, and then the next thing that's going to happen is another tRNA comes in. Oh, excuse me, I made a mistake. So the next thing that's going to happen is the ribosome is going to move over like a conveyor belt. Then the other tRNA comes in, right? So it's going to come in, peptide bond is going to form. Then remember this tRNA, the old one, gonna leave. Bye. And then it's going to shift again. Then this amino acid is going to attach to that one with a peptide bond. This tRNA is going to leave. You guys know the drill. Okay. Keeps going until we end up here at a stop codon, and then, oh, sorry. This one's a stop codon, UAA. And, excuse me, when we hit the stop codon, that's when it stops. Whoa, okay. So you guys have seen this before. The tRNA comes in, blah, 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 peptide bond. Okay, another one comes in. Whoa. Keeps going. Amazing. This is how it works. Once we get to a couple hundred of amino acids under our belt, eventually we're going to come to the stop codon. At that point, we stop translating. And it's going to break off here. This polypeptide right here is going to go over the Golgi, get folded up into a protein. This tRNA is going to leave. This mRNA is going to get broken up into nucleotides, because remember, we don't care about the middleman RNAs. All we care about is the big protein that's being made. Okay, here's a cute picture. You've seen something exactly like this before. Remember that the central dogma of molecular biology is DNA to RNA that's eventually going to make a protein. Remember, this arrow is representing transcription. This arrow is representing translation. So if you look, even in the central dogma, we're admitting RNA is a middleman. We're starting with DNA, using RNA to make a protein. Okay, this right here sums up the steps. It's a pretty nice little flow chart. Just remember, yes, the mRNA goes out into the cytoplasm, but it's important that it ends up at the ribosome, not really that it's in the cytoplasm. Okay, so this is what we're talking about, mutations. We went over this in class. All right. Then we have gene mutations. So there's two different types of mutations. We got gene mutations, and we also have chromosomal mutations. Gene mutations only produce a change in a single gene. So remember, the chromosome looks something like this, and it has bands going across it. They literally, we'll hopefully get to talk about electrophoresis, but there's really bands of DNA that make up genes. We represent them with letters always. Okay, so let's say we have a gene mutation in gene B. Okay, that's fine. Only gene B is going to get affected. In a chromosomal mutation, the whole chromosome is going to be messed up. Okay, so single gene. There are two different types of gene mutations. There's point mutation, which is what we did in class during bell work. So that's where we switch one letter for another one. Make a mistake, basically. But we don't leave anything out. We don't add anything in. We just switch it. Okay, that could cause one amino acid to change. This one changed from gly to seer. Right? But remember, we talked about silent mutations, which could result in no change. Right? And again, it's just placement of the mutation. What's the codon that's being mutated, and which letter? It's all just dependent on which one. Very specific examples. Okay. But the important thing to realize is that in this point mutation, it's only one amino acid that's going to change maximum. On frame shift mutations, however, this is going to change a bunch of amino acids. You see these down here in blue are the ones that have changed. So frame shift mutations, we have insertion and we have deletion. Okay, so obviously insertion is when we're going to insert an extra base. Deletion is when we're going to take one out. So this picture is showing the deletion of this U. So I believe we talked about this in class, but just to make sure, okay, where this codon is supposed to be U, 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 the codon is now U, U, G. Then where the next codon is supposed to be G, G, C, we now have a codon that is G, C, G. And so on and so forth. G, C, A, nope, now it's C, A, U. Okay, and it keeps going like this. So what happened when we got rid of this one U is all the rest of the bases, the nitrogenous bases, the letters, they shifted over to the left one. And remember, ribosome does not have a brain. All it's going to do is read the codons, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. So from here on out, all the amino acids are going to be all messed up. Okay, so the important thing is that a frame shift alters the groupings of codons. It's a great way to say that. Instead of having U, 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 we have U, U, G. And then it's going to keep altering the codons from there on out. If we alter the codons, we're going to alter the amino acids, therefore altering the protein. Okay. All right, so chromosomal mutations. Just get all this on there. There are 4 types, but I will tell you right now, this stuff, I'm not going to make you, we're not making you write it down in your guided notes, okay, the definitions. You just need to know basically the difference between the chromosomal mutation and the gene mutation, and then the fact that these exist, that there's different ways you can have chromosomal mutations. So a test question might be like, hey, right here, part of this chromosome broke off and went and attached to this other chromosome. Is that a gene mutation or a chromosomal mutation? And you would say, oh, chromosomal, duh. Okay, because remember, chromosomal is when you mess with the whole entire chromosome or a whole chunk of it, not just one little rinky-dink gene, right? Okay, so lastly, mutations may or not make an effect. Remember, we have, for may not, we have silent mutations, okay? Sometimes, if you have a real big mutation that ends up in a big phenotype change, okay, it's going to affect an entire ecosystem. Remember we talked about the little duck guy or chicken or whatever he was? There he is. Say hi. Okay, he had 2 wings, but he also had 4 little legs. Now, in some classes I know we got into a little bit of a debate about this. Maybe this could make him run really, really fast. Maybe it would hinder him and he couldn't run that fast. Right? Let's say it makes him run super fast. Now he's able to outrun all his food. Okay? So all his food, if this chick has babies and his babies have 4 legs, right? Now the things that eat the chickens, like what, foxes or hawks or owls or something, they're going to get outrun. These chickens, there's going to be way too many of them. The owls and the foxes are going to be really hungry. They're going to starve and die. Okay, so that's how it can affect an entire ecosystem. That's a very extreme case, but it happens. Okay, and then just remember, not all the genetic information is expressed. We're talking about introns. Okay, not all the DNA does jack diddly. Okay, so that's important to keep in mind. Okay, and again, mutations not always bad. And something important to keep in mind is this right here. Mutations are the reason why we have genetic variability. That means why we have differences between a bacteria and a human being and a fish. Okay, it's all because of mutations that added and added and added over time. Okay, because remember, we all have just 4 letters, A, G, C, T. Okay, the mutations change them, put them in different orders.